Okay, it looks like everyone is coming in. So before we start, I would like to walk you guys through the logistics of our intermediate course for the SynBio4All Africa initiative for this year. A lot of you guys may have known already that we previously has a beginner class last year that we focused on like the simple techniques and simple area of synthetic biology. So if you guys haven't seen the beginner class yet, please feel free to check into our website. You will search SynBio4All Africa in Google. The website should show up, and you can check our recordings and the lecture notes that we have previously prepared for the beginner class. You don't need to go through all that to take our intermediate course, because we make sure our lecture is self-contained. But if you do go through those lectures, it should help you a lot in terms of like getting into the same speed with our lectures that we're preparing throughout this course. Again, before we get started, my name is Ice. I'm at the University of Washington in the United States, and I'm like representing the team at SynBio4All today to welcome you all to SynBio4All Africa. We are having a 10-week intermediate course starting from today, May 1st to the first week of July. And at the last three lectures of our intermediate course here, we will walk you through how to think about the project that would be like related to SynBio, on how to like think through how we formulate the project in a scientific manner. So please stay with us. And that was like something that we are really excited about, and we believe that like you guys will enjoy working with us to develop like an interesting project that might be capable of solving either the local problem in your area or the global problem that the world has been seeing in these past years. So that is what I really want to say, and thanks a lot everyone who are excited about our initiatives and attending the lecture live with us today. I believe there are like a lot more of you who are attending the lecture virtually or like attending afterward. We will make this lecture available very soon, maybe by this weekend, so you can rewatch it as well. And like thanks a lot to the key organizer for this event, Emmanuel, Kato, who is also on the call here, and Harikrishnan, who has organized the meeting for us. Like in this call, you probably see Hedy email going through. And for today, Hari, and his full name is Harikrishnan Namboothiri. Sorry, Hari, if I pronounce it incorrectly. Hari will walk us through the synthetic biology process. And this will be relevant to what has been available in the previous class, in the beginner course that we provided. But Hari will walk us through like the overview of what we have gone through. So even though you haven't watched the whole beginner course yet, I believe Hari will provide us like the important knowledge that you may need to go through the whole class today. Please take it away, Hari.
Thank you, Ice. So my name is Harikrishnan. You can call me Hari. I will be the instructor for this class. This is just an introduction to synthetic biology, a review. So I have compiled some relevant topics from the introduction to synthetic biology webinar that was conducted by SynBio4All Africa in the year 2023, fall of 2023. Yeah, let's get started with it. So here's the outline of my talk today. So I will be talking about what is synthetic biology, give you a brief idea about what do we mean by synthetic biology. Then I will talk about different biomolecules and bioprocesses that the cells use to make proteins. And then I will move on to talk about how does DNA help to control the protein production in the cells. and how to implement all these designs that we talked about in laboratory. So I will talk about some lab techniques in synthetic biology as well. So let's get started. So what is synthetic biology? So by definition, synthetic biology is a field of science that involves redesigning organisms for useful purposes by engineering them to have new abilities. What do we mean by redesigning organisms or engineer them? So it involves understanding how living organisms are organized on a molecular level and what are the types of mechanisms that goes into the biology that we are able to see. Once we understand all those mechanisms, we can use engineering principles to design our own synthetic systems, building upon the existing and making use of the existing natural phenomena already present in the living organisms. And since synthetic biology involves a lot of engineering, it has attracted researchers from different disciplines to use their principles into designing different synthetic systems. And as a collective effort, it has combined the best of all the disciplines that have to offer. So just to give you a bit of a motivation, we'll see an example of what an engineered organism might be like. So biological clocks exist in almost all of the organisms, with some exceptions, and they function by oscillating between different states. So either a protein will be produced or not produced, and it will oscillate between the two states. And naturally, it has been studied in different organisms. And here is a movie that shows a certain protein being made and in an oscillatory manner over a span of few days. And this was observed by using proteins that produce luminescence. Now, this mechanism is not found in a bacteria, let's say E. coli. So using principles of synthetic biology, scientists were able to incorporate certain oscillatory mechanism by using much more simpler mechanisms, by using much more simpler networks of proteins in a bacteria called E. coli. And the oscillatory signals were generated using fluorescent proteins. So if you can see here, the cells are like showing fluorescence, and the proteins are being made and then degraded and things like that. So it looks like it literally looks like the cells are like blinking. So this was one of the initial works in the field of synthetic biology. This system was called as the replicator, and my aim for today is to uh, talk about different concepts, and eventually you will be able to relate how was this particular system designed, and how can you implement that in a lab as well. So the next topic would be talking about the biomolecules and the different bioprocesses that are used by the cells to make proteins. So we all have seen this diagram somewhere or the other. It is the central dogma of molecular biology, which was proposed long back by Francis Crick in 1947. So this tells us about what is the direction of the flow of genetic information in living systems. So what this tells us is that information from DNA is passed on, or it's copied onto RNA, which then takes that information for the cells to make different types of proteins, and it tells the cells what protein to make and when to make. But this particular order of information is not that straightforward, and this is not the complete picture. And over time, we have discovered different processes that might make this particular unidirectional flow look a bit more complicated. So, for example, we now know that DNA also replicates by copying itself into two different copies, which is known as DNA replication. And then we also have the flow of information from DNA to RNA using the RNA polymerase enzyme, which is known as transcription. But it's also observed that the reverse is also true by using a certain enzyme called the reverse transcriptase. And it's also known that the RNA molecule can also be replicated to another RNA molecule by using certain enzymes called RNA-dependent RNA polymerase. And we also know that the the information from RNA can be passed on to make proteins by using a process called translation. So as you can see, right now, this particular central dogma that we had, which was postulated earlier, it has now a lot more arrows in this particular direction of flow of information. So what makes all this possible? What are the different biomolecules and processes that go into these steps that actually ensure that the genetic material is going from DNA, and it is helping the cells to make the different proteins at different times and according to the requirement of the cells? So in any living organism, there are many biomolecules that you find. And out of all those biomolecules, these are the four major biomolecules that you will find, which are the lipids, nucleic acid, carbohydrates, and proteins. And since we are interested in knowing how the cells make protein, how genes are being expressed to make the proteins, the two most important biomolecules that are involved in that particular process are the nucleic acid and proteins. So I will talk a bit about nucleic acid and proteins in a bit more detail. So what are nucleic acids? So they are the molecules that actually carry the genetic information. Now, nucleic acid is made up of smaller building blocks called nucleotides, which have a structure as shown here. Stop that, please. Okay. So there is a sugar molecule called ribose, which is a 5-carbon sugar molecule in the center. And we have a base, which is a nitrogen base. And we also have a phosphate group. And all three of them together, they are called as the nucleotides. And depending on the type of base that is present, or depending on type of the sugar that is present, the nucleic acids can be divided into different categories. Now, this nucleotide can form different chains of nucleic acids, nucleotides, which form different type of nucleic acids. So as you can see, the phosphate group, it forms like the backbone of the chain of nucleotides that it makes. And we have different kinds of bases that can be attached to the nucleotides, which can be broadly divided into two groups, that is purine bases and pyrimidine bases. The purine bases, as you can see, will have like two adjacent rings, like hetero cyclic rings. And we also have pyrimidine bases, which are like single rings with carbon and nitrogen both in it. And there is also another type of base, which is also a pyrimidine base, which is called uracil, which is only found in RNA and it's not found in DNA. And I will talk about what's the more differences between RNA and DNA in the next slide. So this is the basic structure of any nucleic acid that you will find, which carry the genetic information. So what is the difference between DNA and RNA? So DNA is a double-stranded molecule. The DNA stands for deoxyribonucleic acid. And so if you see the difference between the sugar molecule in the DNA and RNA is that the presence of the OH group here, and there's no OH here. That's why this is called the deoxyribonucleic acid. And another difference is that the base that is being used in DNA, which is thymine, it's not present in RNA. We see another type of base called uracil, which the only difference between them, these two molecules, is the presence of the methyl group here. And then the other bases are this cytosine, guanine, and adenine. And you can see that DNA is a double-stranded molecule. So what are these base pairs? So at different sites of DNA, if we depending on the base that we have, So let's say we have cytosine, then cytosine can form three hydrogen bonds with guanine, and adenine can form two hydrogen bonds with thymine. And that's how these base pairs are formed. So these base pairs are nothing but hydrogen bonding between the two bases on the inside of the long chains of nucleotides. But RNA is a single-stranded molecule, so we don't see that. Usually RNA is single-stranded, so we don't see that the hydrogen bonding often. And then the second type of biomolecule that I'm going to talk about is proteins. So proteins are they can be they can be called as the molecular machinery of the living systems, but they carry out a plethora of functions in the cells. So the structure of protein is something like this. So amino acids form the building block of every protein. So this is the structure, basic structure of an amino acid. So we will have a carboxyl group, we will have a amino group, and then we will have a side chain, the R. It can be anything from a methyl group or any other hydrocarbon group. So in human body and most of the organisms, naturally we see about 20 different types of amino acids, although more amino acids have been discovered. So each amino acid, they can form a peptide bond with other amino acid, and a chain of amino acid, it's called polypeptides. And this polypeptide can fold into different configurations, leading to different structures of proteins. So these are the different structures of protein. First one is the primary structure of the protein, which is nothing but the sequence of different amino acids. And this is just visualized as a bead of all the amino acids just forming the polypeptide chain. And then through different interactions and through the hydrogen bond or different types of molecular interactions between the different side chains of the amino acids, the protein primary structure can be developed into two types of different secondary protein structures, which is the alpha helix or the plated sheets. And then a protein can have both plated sheets and alpha helix in its structure, and both these structures will then interact and with different molecular interactions, and with the interaction with the media or surroundings, the protein can then fold into a tertiary protein structure, which may contain both alpha helix and plated sheets, or just one of them, or like something like that. And then we also have another layer of structure called the quaternary protein structure, where it could be a complex of two different structures of protein, which are formed from different amino acid chains. So depending on the structure, uh, proteins can have different types of functions. So they could be just having function to maintain structural integrity in the of the cells. For example, if we have proteins such as collagen or keratin, that they have a function of maintaining the structural stability. But we also have proteins that can carry out that can catalyze different biochemical reactions, which are known as enzymes. For example, we have trypsin or hemoglobin or proteins like that that can actually carry out different reactions, biochemical reactions in the body or in the living cells. And then these biomolecules can be used to carry out different bioprocesses in the cells. So first one of them is DNA replication. So how cells copy the genetic information. So DNA replication is very important for cells because when the cells divide, it needs to divide in such a way that each of the daughter cells will have the DNA so that it can carry out the further biochemical processes using the DNA, because DNA is the molecule that carries the genetic information. So DNA replication becomes a very important process in which the genetic information is first copied and transferred into different cells, the daughter cells. So there are many biomolecules that are involved in DNA replication. So this is the simplified version. Like this is how DNA replication can be visualized. But in reality, we have different enzymes that make it happen. So we have helicases, which are large proteins that unwind the double helix structure of the DNA, and it will let the DNA polymerase to actually carry out the replication process. It will help the DNA polymerase to build onto the DNA strand. And then we have single-stranded binding proteins that actually stabilize the unbounded DNA. And DNA replication will happen on both the strands, the strand from 3' to 5', and also from the 5' to 3'. But it should be noted that DNA synthesis, the new strand synthesis always happens from the direction of 5' to 3'. So what happens for the other strand is that since the elongation can only happen from 5' to 3' direction, here the new strand is made in this direction. But if you see at this is called the lagging strand. So if you see, the direction of elongation is opposite to it. So that's why this for the lagging strand, the elongation is very discontinuous. So that leads to formation of something called as the Okazaki fragments. And later on, this discontinuity is eliminated by using another enzyme called DNA ligase, which can join all the gaps that are there in the elongated DNA. So this was how different enzymes help in DNA replication. Uh, so far, does it? Okay. So the next process is transcription. So now we know how DNA is being replicated in the cells. Now, how does the cell use this DNA to transfer information onto RNA that can actually let the cells know what proteins to make? Or so how is this process being done? So this is a schematic showing that how DNA RNA can be synthesized using DNA as a template. But how do enzymes carry out these processes? So we have an enzyme called RNA polymerase that can bind to certain part of the DNA called the promoter, and it will form the promoter complex, which moves from a closed configuration to an open configuration where it opens the that particular section of DNA, and it can also unwind the DNA, and it starts to read the DNA from that particular point. So that step is called the initiation step. And once the transcription has been initiated, the RNA polymerase will start to elongate and go on creating the RNA transcript that is also made in the 5' to 3' direction. And then finally, the process of transcription ends at a place where we have the terminator. And at that point, the RNA polymerase will break free from the DNA template. And this process it happens at a very fast rate. Like it happens at almost a rate of around 30 to 40 nucleotides per second. So the RNA polymerase can make mRNA strand at that particular speed. So these molecular machines are very efficient in carrying out these complicated steps in a very at a very fast rate. And then once we have the mRNA molecules, mRNA strands, how does the cell make the protein using that particular mRNA strand? So now since we have the mRNA sequence will contain certain sequences called codons. So each codon, which is a set of three nucleotides, it will code for a certain amino acid. And then all those amino acids will form the polypeptide chain, which further folds into a protein. So this whole step of translating the mRNA sequence into the language of amino acids is called translation. And this is carried out by another molecular machine called the ribosome. So ribosome is a complex, can be called as a complex of two different proteins. It is formed using two subunits of ribosomes. The smaller subunit, it is responsible for binding to certain sites on the mRNA. And the larger subunit is responsible for elongating or making the polypeptide chain as it travels through the mRNA sequences. This step also requires the presence of another nucleic acid called the transfer RNA. So the transfer RNA is responsible for bringing the amino acids from the pool of amino acids to the ribosome. And the ribosome has the ability to detect what is the codon, and corresponding to that, the tRNA will have the anticodons, and it will recognize what tRNA, which tRNA should be included for the process of translation. And the tRNA will have the amino acid already present on it. And then the ribosome will further catalyze the step of adding this particular amino acid onto the polypeptide chain by forming the peptide bond. And the direction of translation, it happens from the 5' to the 3' end of the mRNA. Okay, does anybody have any questions till now?
Like it's possible to type into the chat as well, and maybe like some of us here can help like discuss with each other.
Okay. So I have a question for you guys now. So since you don't have a question, I will ask you a question. Why do you think organisms have evolved to have an extra step of making mRNA and not just make proteins directly from DNA? Because it's basically taking the same sequence that is replicated again and again. So why not just directly synthesize the proteins using the sequences from DNA? Can anyone answer that? Okay, we have question.
I see Habib Mana raise a hand. Do you want to answer? You can unmute yourself and talk if you'd like to.
I think it's because the DNA can't leave the nucleus. Hello?
Yeah.
Yes, your question. I think it's because the DNA can't leave the nucleus.
Okay. In the case of
So the information in the
nucleus. So what will happen in the case of a prokaryotic cell where everything happens inside? There's no defined nucleus.
okay. I don't know.
Can I try?
Maybe.
Can I try?
Yes, yes.
One, it is the tightly packing of DNA, which if it keeps on unwinding, then it means that we'll be using so much energy. Then two, due to that, the process becomes inefficient. There is also another bit about the ribosomes in the cytoplasm, where the process occurs. The DNA cannot be moving in that process as such. So those are the two areas where I feel there'll be an issue.
Excuse.
We have more people.
Excuse.
Okay. Yeah, all the answers which everyone gave like kind of they direct toward so that you don't have to depend on the DNA all the
Yes.
time, make new proteins again and again, and you can make more mRNA, and you can make more protein from each mRNA every time. So you don't have to rely on the DNA sequence that is like the original copy for making proteins all the time. So this is a very smart way of creating a temporary memory type of thing in the cells, so that the proteins can be again and again made from the mRNA strands, and you don't have to refer to the DNA sequence all the time you need to make protein. And yeah, there are many other factors as well, and everyone has listed, I think, uh, different correct answers in the chat as well. Okay. Does anyone have any other questions? You can raise your hand and ask unmuting.
Hi.
Hi.
Yeah, so thank you for the presentation. And I just needed some bit of clarification about the Okazaki fragments. So if I understood you right, these still happen in the 5-3 direction.
Yeah.
Smaller scale in smaller pieces, and then they're joined together later by the ligase. Is that so?
Yes.
Okay, how is it possible for them to happen in those smaller at those smaller scales? Like, what is the mechanism behind that? Why doesn't it happen? Why don't we have longer strands? Why are they short? Why should they be short? What mechanism facilitates that restriction?
Yeah, that's a good question. So when the elongation, like when the DNA polymerase is making the leading strand, the complement of the leading strand, it has a starting point, and as and when the polymerase is moving ahead, the replication fork is get opening like as and when the polymerase is moving. But it's not true for the lagging strand. So the DNA polymerase needs a starting point, a primer, to actually start the process. So we have an enzyme called primase that can actually get that starting point for that DNA polymerase, and it has to start from the 3 prime and go travel to the 5 prime and make the DNA from 5 prime to the 3 prime direction. So as and when that is happening, if you can see that this replication fork that also like it travels ahead, and that's the reason why the lagging strand the synthesis of the lagging strand is very discontinuous, because you need to make primers as and when the replication fork is going ahead. So that's why this process of synthesizing the lagging strand is discontinuous. And since it is discontinuous, that's the reason why we have the shorter fragments made again and again. Because we don't see that fragments in the leading strand because it has only one starting point, and it can go on synthesizing the leading strand as and when the DNA is being opened.
Okay, thank you.
Excuse, I have a question. Excuse.
Please.
Okay, thank you for the presentation, which has been very insightful. The question that I have is, I have two questions, but the other one is, okay, hypothetical one, but okay. The first one is, in stage 2 of replication process, indicates that after the unwinding process by the helicase, we have the single-stranded proteins that binds and stabilize the unwound parental DNA. So I wanted to know the examples of these proteins and okay, the examples of those single-stranded proteins that helped in that process. And then the second question is, it's a hypothetical one. We can see that that the replication process starts from the 5' end to the 3' end. And then also the synthesis of the Okazaki fragments. So my question is, can there be some instances or rather abnormalities? Now, that's why I call it hypothetical. Where the synthesis sometimes if the process goes wrong in the cell, that can make the process to start from the 3'. And if there is such kind of cases, maybe because of some abnormalities, then what can that result to? Yes, thank you.
Okay. If I understood your question correctly, you want like an example of the single-stranded binding protein, which are like bound to the DNA. So I'm not aware of like specific examples of like what are the names of those binding proteins. But yeah, I will get back to you if I get like any specific examples of those proteins that help in stabilizing the unbounded parental DNA. And to answer your second question, the synthesis of the strand, it cannot happen from 3' to 5' direction, because that is a very like the energy required for that will be much higher than what it requires for the synthesis from 5' to 3' end. So that the answer to that lies in the chemistry of the each nucleotide being added after each nucleotide. So the energy required to form the new bonds after the hydrolysis is like I think it's lesser. It's more energetically favorable for the cells to synthesize new fragments from 5' to 3'. That's why it has evolved. The cells have evolved to do the enzymes have evolved to do that without the requirement of the nucleotides having to bring the like the I think the yeah if I yeah I'm also not sure exactly why would the cells all the enzymes evolve to do that from the 3' to 5' direction. But yeah, I'm not sure if I have answered your question.
Oh, yes, you have. Thank you.
Okay, there's one more question. Is single-stranded protein same as topoisomerase? So, no. So I think there are two different enzymes. So topoisomerase is an enzyme that can so the DNA is found in a very coiled manner. So the topoisomerase helps in removing all those coils and actually changing the topology of the DNA strand such that it's much more easier for the enzymes to carry out the DNA replication, because it is not possible when the DNA is super coiled, like highly coiled and condensed state. Yeah, if you have any more questions, I think we can discuss them in Slack as well. You can ask them in Slack, and I will be more than happy to discuss it with you. For any more questions? There are some questions that I think will be answered over the next few slides. Okay, so I think we can move ahead. So the next part that I'm going to talk about is that how does the different sequences in the DNA help the cell to decide how much protein to make or what protein to make, when to make, and things like that. So DNA, we know that it contains all the genetic information to make specific proteins, but at the same time, making the proteins are very energetically expensive for the cells. So one can imagine that if the synthesis of protein happens for all the genes at all the time, then we will have production of mRNA all the time. We will have all the ribosomes engaged in making proteins for all the mRNAs, which is not favorable for the cell because it takes a lot of resources to make proteins. And in the case of multicellular organisms, where different types of cells at different parts of the organism might require to make different types of protein, which give them their specific purpose in that organism. So that won't be possible if all the cells are making all the proteins at the same time. And also, so because of that, we have different regulatory components in DNA that help to the cell to decide what protein to make based on maybe certain indicators, and how much protein to make based on the interaction between the DNA or the mRNA between the different enzymes present. So I will talk about each of those parts on the DNA sequence. Okay. So how in any synthetic biology like literature, this is the common like way to show the different parts on the DNA. So we have a promoter, and then we have the RBS, which is the ribosome binding site, and then we have CDS, which is the coding sequence for the protein, and then we have a terminator. And each part, each sequences on the DNA will have its own impact on expression of different genes. They can affect how much protein is being made. I think that was one of the questions. So there are different these are the different parts or the sequences on the DNA that can actually determine how much protein is being made, or if at all the cell needs to make the protein or not. And engineering these different parts by carefully optimizing the sequences for all these parts is what we can call as the tuning of different parts in synthetic biology terms, to engineer how much protein is being made or what type of protein is being made based on different inducers or different chemicals that can actually induce, or we can also engineer the different parts to be more attracted, or like increase the probability of expression of genes. So I will talk about each of the parts in a bit more detail now. So promoter is a region in the DNA where the RNA polymerase actually binds to initiate the transcription. So the sequence of the promoter, it actually decides what is the strength of the interaction between the DNA and the RNA polymerase enzyme. And the greater the interaction, the stronger the interaction, the more probability we have that the RNA polymerase will bind to that particular DNA sequence. If we don't have a stronger interaction, we will have a lower probability of the RNA polymerase binding to that particular DNA sequence. And once the RNA polymerase is bound to that sequence, then the promoter will decide how much RNA is being made and at what rate. Because the sequence of the promoter can also affect the rate at which the RNA polymerase is binding to that particular sequence and initiating the transcription. So we have, broadly speaking, we can have two different types of promoters. One which is constitutive promoter, which is always expressing, always facilitating the production of mRNA. And then we have something called the inducible promoters. So one interesting property of the promoter is that it can have a certain sequence called the operator, where certain molecules called repressors, which are usually proteins, can bind to that particular site, and it can inhibit the transcription from that particular sequence. So the probability of making mRNA from using the RNA polymerase will decrease significantly when we have an operator region where a repressor can bind. So then using that property, we have inducible promoters that usually work by binding to the repressor and just taking away the power of the repressor to bind to that particular site and inhibit the transcription. And there are other mechanisms as well that can be used to create an inducible promoter. So when a promoter is induced, that means that we have provided certain input signals for the promoter to be activated or not activated, and then further drive the gene expression. Then we have the RBS, which is the ribosome binding site on the mRNA. So this sequence actually decides how strong the interaction is between the ribosome and the mRNA. And the ribosome needs this RBS to recognize the site at which the ribosome can bind to and later on start the translation. So based on different types of RBSs that have been engineered, that can also decide what is the expression level of different proteins based on different RBS. So it's a common practice to generate like a library of RBS with different sequences and see how the production of the proteins change with different RBS sequences being used. And then we have the CDS, which is the coding sequence for the gene of interest. So the CDS usually starts from the start codon, which is ATG, and then it ends at the stop codon. We have three different types of stop codon. Any of them can be used to stop the translation at that particular point. And usually CDS are optimized in such a way that the codons that are present in the CDS that are available, that are abundant in that particular organism that we are interested in. So that's why usually when we deal with CDSs in different organisms, we also need to optimize the CDS such that the codon is optimized for that particular organism. And then we have the terminator on the in that terminator part that is responsible for stopping the transcription, and that's where the RNA polymerase will stop transcribing the DNA. So it usually is a form of a hairpin loop, and it's usually rich with the nucleotide uracil. So it's also okay. So I will ask you the question. So I have a question for everyone. So if we are using a stop codon in the CDS, then why do we require to use the terminator. Yes. Nelson, you raised your hand. You can answer.
I think terminator is to stop the transcription process. The function, I mean.
Yes. Okay, I will repeat the question. So if we have a stop codon in the CDS, then is it fine if we don't have the terminator? Because the protein will anyway stop, the ribosome will anyway stop making the protein once it reaches the stop codon. Yeah, Joy, yeah, you can go ahead.
Okay, thank you. I guess the terminator is used in the DNA while making of the mRNA, and the CDS is used in the protein making. So terminator is important in the DNA and the CDS is important in the protein making.
Okay. We have more answers.
Maybe.
Yeah, either any one of you can go. I think
Maybe.
Yeah.
Maybe with the terminator, it helps stop the whole process, like to stop the transcription process, so that we can have that we can have the mRNA that has been made get released.
Yeah. Yes. Anybody else want to add in?
Let me comment on the terminator.
Yeah, sure. Go ahead.
Yeah, so majorly the terminator is to knock off the enzyme from the sequence of the genetic material rather than allow it to continue to move along the sequence.
Okay. Okay.
Also, the terminator will signal the end of transcription, whilst the stop codon signals the end of translation.
Yeah.
I can also add something.
Go ahead.
So if the terminator wasn't there, that newly synthesized protein would still remain attached to the ribosome, the ribosomal machinery. So yes, the process would end, but then the protein wouldn't really be released.
I'm not sure about that. Okay, the terminator is for the RNA polymerase to stop making the transcript, but the stop codon is recognized by the ribosome to stop like making more of the protein. It will just stop making proteins there. So after that, the protein is released off of the ribosome. But if the terminator will ensure that the mRNA synthesis is actually stopping there. And the codon will ensure that the protein synthesis is stopping there. So if we don't have the terminator, and if we have some other gene downstream of that particular gene, then the RNA polymerase does not have enough information to stop the transcription of the required mRNA, and it can go ahead and make the transcript for different genes as well. So for specific, if we want specific transcripts, if we want specific mRNA to be made, then we need to add the terminator so that the process of transcription stops there. And the stop codon is added in the end of the CDS so that the protein translation of the protein stops there by the ribosome. Does anybody have any questions?
I have a question.
Hello. Hello.
Go ahead.
Can I talk? This is Gloria. Yeah, I was going to say about stop codon and terminator, you could be using constitutive, promoter, so you have more than a single gene, or a series of proteins being produced at the same time. So stop codon can allow you to have truncated segments of what you want to produce, but with terminator, you're completely stopping the whole process, which you have just.
Yeah. Gloria, you had a question?
No, it wasn't a question. I was just trying to add on to stop codon in relation to terminator region. I'm like, if you're using constitutive promoters, you may have more than, a single gene you want to translate. So you have, you have transcription happening more than just once. So a stop codon may not really limit the RNA polymerase from, transcription from taking place. But if you have a terminator, automatically the polymerase will stop whatever action it's doing. So it's much more less predetermined than having stop codons. You can have stop codons truncated between, in between segments you want to, transcribe. Thank you.
We can have that conversation on Slack as well. If you want to add anything to that, you can message any one of us on Slack, and we can discuss that in more detail on Slack. I think there was one more question.
Yeah, I would like to add one thing to that, Harry. I'm creating lecture question channel in Slack, so you please feel free to drop that publicly, and not just us, but you guys can share your thought on these kind of questions as well. That should help a lot with our ongoing case question here. Yes, thanks.
Is there any more questions?
I have a question.
Yeah.
So, what is the necessity evolutionary per se to have 3 stop codons? Why not restrict it to 1 stop codon so it makes stuff easier, faster, and so on? Is there any reason, or is it a consequence of?
That's interesting. I'm not sure about that. Why the cells have evolved to have multiple stop codons. I don't have an answer right now. Sorry. We can.
I have a question too. So when it comes to the terminator, what exactly happens? How does it stop the polymerase? Is it due to is it a physical thing because of the hairpin, that the hairpin somehow obstructs or is complex to navigate, so somehow the polymerase just has to give up and stop doing the thing and then it terminates there?
There are two reasons. So like one of them is, yes, the hairpin, like it obstructs for the polymerase to further carry out the transcription, and also it's also the region is filled with U nucleotides, uracils. So that also has to do with reducing the affinity between that particular region and the RNA polymerase. So there are different reasons why the terminator can actually terminate the process of transcription.
Thank you.
Okay. Okay. So we can, I will move on to the next slide. So once we have all these parts, how do we express these parts in a bacteria, let's say? Then what do we use is something called a plasmid, where all these different DNA parts can be assembled together and then transformed into a bacteria or any other organism of our interest. So plasmids are also circular DNA, but they are not the same as the chromosome of that particular bacteria, which actually contains the genome of the organism. It's different from that. It's a circular DNA that can replicate whenever the cells are replicating, and it will have enough sites for our synthetic parts to be incorporated in. There are some of the important parts of a plasmid are the origin of replication. So the origin of replication will ensure that the plasmids in the cells are also divided as and when the cells are dividing, and they are replicated into each replicated and divided into each of the daughter cells, so that all the cells will contain the plasmids with our gene of interest in it. And the origin of replication will also determine how how much copies of that particular plasmid will be replicated in the cell. So based on that, we can have a low copy plasmid or a high copy number plasmid, where we can have anywhere from 5 to 10 plasmids in the cell if it is a low copy plasmid, or we could even have like 100 to 150 copies of plasmids if it is a high copy plasmid. So the probability of replicating that plasmid increases based on the origin of replication that we use. And it is very important to have the origin of replication, because without that, the plasmid will not serve its purpose at all. And then if we are using multiple plasmids in a single organism, then it's also necessary to consider the origin of replication, because we need compatible plasmids that are compatible with each other and the host, so that the origin of replication can actually ensure that the plasmids are replicated when the cells divide. And then another important part of a plasmid is the selectable marker. So usually we have an antibiotic resistance gene that help us to select that act as a marker in the plasmid. So why do we need a selectable marker? So whenever we transport that particular plasmid inside a cell, the selectable marker helps us to identify those cells that actually took the plasmid in, and they are expressing the genes inside the plasmid or not. So if we have an antibiotic resistance gene, let's say the gene is responsible for building the resistance towards the antibiotic ampicillin, then this gene will be responsible for making the proteins that make the cell resistant to the antibiotic ampicillin. And if we have ampicillin in the media, the cells will be able to grow even with the presence of ampicillin, which is an antibiotic. But the cells which don't have that resistance gene, or the cells that did not take up the plasmid during the process of transformation, will not be able to grow in the media with that antibiotic because they don't have this plasmid which contains the antibiotic resistance gene. So that's one way to selectively screen for which of the cells have actually taken up the plasmid. And also it should also have multiple cloning sites. I will talk about that in one of the later slides as well. So we need a site on the plasmid where we can actually cut the plasmid and insert our gene of interest. So that is also a necessity, naturally a necessity in a plasmid, because without those sites, we won't be able to insert our desired fragment with our gene of interest in a plasmid. So, based on all of the notations that we saw, and how does each of the promoter works, how does the CDS work, how does a repressor can inhibit the transcription, based on that, the movie that I showed you in the beginning slide, it actually is from a bacteria that actually contains these two plasmids. So this is called the repressolator. So what, as the name suggests, how it is designed is that each of the protein, there are three genes that code for three proteins, and each protein is responsible for repressing another production of another protein, because this protein acts as a repressor for this particular promoter. And another protein will act as the repressor for another promoter, and that will repress the production of a different protein. And that protein will also in turn be repressing the production of the protein that is before that protein, which is the tetar. So each of the production of the protein will inhibit the production of other proteins. So this actually forms a system that can oscillate. And the concentration of these proteins can oscillate, and we have a reporter gene, GFP, that actually tells us how the concentration of one of the proteins is oscillating. So that is how we can observe the oscillations in the cell. I'm not able to play this movie. Oh, okay. Sorry, this movie is not playing. It's the same movie with the bacteria showing oscillations in the fluorescence. So also, it is also possible that all the concentrations of these molecules just reach a constant concentration and they don't oscillate at all. So it was actually derived using mathematical models and using dynamical systems analysis to show that what kind of promoter we need, or like how strong should the repressor bind to the promoter, or how fast should the proteins degrade in order to like match the lifetime of the mRNA. And a lot of analysis like that led to the design of this particular construct. And because of that, synthetic biology as a field, it is using engineering principles to design these gene circuits, and that's why synthetic biology emerged as actually an interdisciplinary field where we can actually use engineering principles, mathematical models to guide the design of different genetic circuits. And the next important thing while designing these circuits is how do you select the host for what purposes? So if we look at prokaryotic cells, they are usually they grow fast as compared to the eukaryotic cells, and the requirements are also usually much lower than what we require for eukaryotic cells. And it is simple to study the genome of the prokaryotic cells, as they are they contain usually lesser genes, and it's more easy to study the genome as well. But eukaryotic cells also contain like the machinery, the enzymes that can actually help in synthesis of much more complex molecules, which the prokaryotic cells might not contain. And also, the eukaryotic cells have a defined nucleus in which the DNA is present, and a transcription happens inside the inside the nucleus, and the RNA is transported out, and all the translation process happen outside the nucleus. But in prokaryotic cells, that boundary is not defined, and most of the time, as and when the mRNA is being made, simultaneously it's also translated. So we have a co-translation happening in the prokaryotic cells. And because of the ease of handling of the prokaryotic cells and how fast they grow, they are more commonly used in bulk manufacturing of synthetic molecules. They are much more frequently used in industrial productions. And eukaryotic cells are mostly used for producing proteins that are much more complex and cannot be created by the prokaryotic cells. So, now we come to how do we implement these designs in a laboratory. So before moving on, does anyone have any question?
Like, would it be possible if you could give like a small intro on like how they did all that? Like you said, mRNA degradation, how the replicator works, like all that stuff. Could you give a small intro on how that is done? Like what is being used to do that mathematically and so on?
Okay, I don't have the slides on that, but what was actually done is that so the gene expression can be modeled using ordinary differential equations, and they are modeled basically in like a simple manner where the rate of production of protein is is a first order rate at which mRNA is being the concentration of mRNA depending on the concentration of mRNA, and the degradation of the proteins are first order degradation reactions, and the mRNA is being made at a constant rate, and it's also degraded at a constant rate. And as the cells, as the repressor proteins are being made, that will also inhibit the rate at which the mRNA is being made. So using all these interactions, we can design ordinary differential equations. And using those ordinary differential equations, we can determine what parameters can actually lead to sustained oscillations in the system. So it's a bit difficult to explain it like just right away. But if you want, I can explain it in more detail on Slack.
Yeah, okay, I understand. Yeah.
Do you have any other questions?
Hello, yes, I have a question. When you are talking about the genetic mode of action, when you're talking about the plasmid, and you talked about there are two types of plasmids that they are found in the cell, you said some, I can't remember the name. Yeah, the replicator. Yes, that one. And you said that it's inhibiting the other genes from the promoter from expressing the protein. Are you referring to the inactivation mechanism where you insert another gene to inactivate the other gene being produced?
No,
yes.
No. Yeah, so good question. So by repression, what I meant is, for example, the protein TET-R is being produced. And TET-R is a protein that will bind to the operator region of the PL-TET promoter. So when the protein will bind to that particular operator region, that will block the transcription of this particular like the whole sequence of all the parts in this for this gene completely. So when the protein is bound to, when we have more protein, we will have more proteins that are bound to this particular operator region, and that will stop the transcription itself. So that's how it represses the production of another protein, and that's the same for all other proteins as well. That's why they are color coded as well. So this yellow TET-R, it will like bind to this particular promoter's operator region, stopping the transcription. And in a similar way, LAC-I protein can bind to the operator region of PLAC. So that stops the transcription of this particular protein itself. So when this protein is bound to the operator region of this promoter, it will repress the production of TET-R protein. And since that particular protein is being made at a lesser rate, it will repress the production of this protein at a lesser rate. Is that clear?
Yes, it is clear. Thank you.
Okay, so moving on to how to implement these designs in a lab. So the first tool that most processes uses the restriction enzymes. So I will start off with okay, so I'll start off with like a story. So let's say I have a particular gene of interest which I want to insert in a plasmid and express that in E. coli, let's say. So how do we go about that particular process? Okay. Can annotate. Okay, please don't do that. So yeah, so we can use restriction enzymes to cut the DNA sequences at particular like our desired sequences, and we can isolate that particular part of DNA from, let's say, a donor plasmid which has the gene of interest that we are looking for. And the restriction enzymes have a particular property such that when it cuts the sequences, it leaves certain overhangs that can be that can recognize other overhang, and the part that we cut here, it will have a certain overhang that will only recognize this particular sequence of DNA which was cut by the same restriction enzyme. So when we use a restriction enzyme to cut the plasmids at different parts, we should use the same restriction enzyme to cut the DNA sequence from the donor plasmid as well. And then finally, we can ligate that particular part into the recipient plasmid and have the assembled plasmids ready. But this process has been made much more easier by using a different type of restriction enzyme, particular type of restriction enzyme called type 2S restriction enzymes, which has a special way of creating these overhangs. So if you notice here, for the BSA, let's talk about BSA1 restriction enzyme. So GAGACC, this particular sequence is recognized by the BSA1 restriction enzyme. So what it does is it leaves one nucleotide and then it cleaves in such a manner that the 4 nucleotides after that particular skipped nucleotide is remaining in the overhang. And if we add this particular BSA1 recognition site on both sides of the part that we want to cut, and we design this particular 4 sequences in a certain way, we can actually create different parts that have specific overhangs that can lock onto different specific overhangs. So in this case, we have 2 fragments A and B that have been cut using the BSA1 restriction enzyme, and it will have an overhang, let's say this orange sequence, And in the destination vector as well, we had the BSA1 site, and that will also leave a particular overhang, which is like in this case orange in color. And these two overhangs will be complementary to each other. And in the same way, A has been cut with a particular overhang in this color, in this case, color coded as red color. And B, the fragment B also will have an overhang that is complementary to this particular overhang. And in the same way, B will have an overhang that is complementary to the overhang in the destination vector. And all these fragments can be then ligated together using a particular enzyme called DNA ligase. So this process is called Golden Gate assembly, where we create different fragments that are compatible with each other, and then we assemble them using DNA ligase. So this process is very commonly used to create fragments with multiple parts, as we discussed before, the different DNA parts. And one commonly used strategy is called the MOCLO strategy, which is the modular cloning strategy. So how that works is that we will have different promoter sequences. We can have different UTRs, RBSs. We can have different CDSs or terminators, which will have the same type of overhangs. So all the promoters will have same set of overhangs, which is GGAG. We can call it the A overhang. And then all the promoters will also have another overhang on the other side, which is TACT, which we can call the B overhang. And all the RBSs will also have the B overhang on the 3' site. Sorry, the 5' site, and the all the AATG, the C overhangs on the other side. So when we create the sequences in this way, and then we use the restriction enzyme BSA1, we can create certain overhangs that are complementary to each other, and then assemble all these parts together. So if we make the promoters, the UTRs or the CDSs, terminators, signaling peptide sequences with the appropriate overhangs while designing itself, we can create a library of promoters and all these parts, and we can mix and match between any of these parts and create fragment of our interest very easily. There is another method for assembling the parts, which is called the Gibson assembly. So that uses the property of a certain endonuclease enzyme that can cleave from the 5' end of the particular fragment and create overhangs. So what people have done is that they have used the same property to create overhangs on different gene fragments, and designing the fragments in such a way that the overhangs created on the 5' end are complementary to each other. They can assemble different fragments onto a plasmid. And this is a very robust technique to actually insert fragments onto a plasmid. So usually fragments are made, so let's say A and B will be made together using Golden Gate assembly. And then Gibson assembly is used to drop these fragments onto a plasmid. And the two processes together, it's called a 3G assembly, where we use the Golden Gate as well as the Gibson assembly to assemble all the parts onto a DNA. And then the next step, now we have the plasmid. Then what do we do with that particular plasmid? Once we before that, we also need to ensure that we have the fragments that are correct fragments that we want to insert in the into the plasmid. So for that, what we do is something called PCR, where we can amplify the concentration of that particular fragment from a few nanograms to micrograms. That's so what? How do we do that? So let's say we have a DNA template of and this is the sequence of our interest, and we will have primers that can selectively bind to that particular sequence. And PCR is carried out with three steps, major steps, and carried out in the cycles of carried out in cycles. So the first step is denaturation, where the denaturation happens at around 95 to 98 degree Celsius, where both the DNA strands are like completely separated out. And the next step is annealing, where the primers will bind to that particular sequence on the DNA. And the next step is extension, where the DNA polymerase can actually synthesize the new molecule of DNA as a new strand of DNA onto that particular required strand of interest. The annealing temperature happens based on the melting temperature of the primers. So that is at what temperature will the primer get detached from the sequence. So usually the annealing temperature is less than like about 3 to 5 degrees Celsius less than the melting temperature of the primers. But that really depends on what kind of polymerases we are using, and depending on that, we can choose the annealing temperature. And the extension temperature usually is around 72 degrees Celsius. That's where it's the optimum temperature for the polymerase to carry out the extension as well. And depending on the size of the fragment, usually for 1000 base pairs, we give about 1 minute for it to extend. And we can tune all these parameters to get the optimum results. And now that we have amplified all these molecules, all these DNA sequences using PCR, how do we separate out the specific sequences that we require? So for that, we use a different method called gel electrophoresis, which is separating the DNA based on their weight. So DNA has a negative charge. And if we pass the DNA through an electric field, it will move because of its negative charge. It will move towards the positive electrode. And we use an agarose gel matrix, and the DNA travels through that particular matrix. And based on the size of the DNA, if it is bigger, it is more difficult for the DNA to travel through the matrix. And the smaller the DNA is, it's more easier for it to travel through the matrix. So I have a question for everyone. So if I do a gel electrophoresis with two DNA samples, and one of the samples is near the negative electrode and the other sample is near the positive electrode, then which of the sample is having like the higher which of the sample is bigger than the other?
Yes,
The
I
one,
think I
yeah,
have
the
a response.
positive is smaller.
Sorry, I couldn't hear that.
The DNA which is near to the positive electrode will be the shortest or the smallest.
Yeah, no, sorry. So the one that has traveled the farthest towards the positive electrode, that will be the smallest DNA among the two. Yes, I think everyone got that right. Okay. So now we have assembled our DNA onto a plasmid. Then the next step that we do is to transform that particular plasmid into the organism of our interest. And there are different methods to do that, ranging from chemical transformation to physical transformation, electroporation, and many other techniques. Out of them, commonly, what I've observed is that people use chemical transformation or electroporation to do this particular transformation. So how this is done is by giving certain shocks to the cells so that their walls open up to receive the plasmid into the cells. And then those cells are rescued in certain media, and then they are plated onto agarose plate, which contain the media as well, and the antibiotic to selectively grow the cells that receive the plasmids. And once we have grown those cells, and we also see the cells growing on the plates, even when we added the antibiotics, that could mean that our plasmid has entered the cell and the cells have accepted that plasmid. But it still does not give us a confirmation whether the plasmid inside the cell has the correct sequence that we want or not. So for that, what we do is we extract the plasmids and we do a sequencing on that. So the sequencing is done using a simple mechanism where they use certain types of nucleotides where which if that nucleotide is being attached to the strand of the DNA polymerase elongation step, it will terminate there itself. So based on that particular property, we will have the termination of the DNA polymerase elongation at different nucleotide of the DNA, and then we will have an array of all the possible combinations of all the possible sequences combinations in that particular DNA sequence. And then all those sequences are either they use like electric field or certain capillary gel to separate out all those fragments. And based on those sizes of the fragment, we can tell that what is the sequence of the particular DNA that we are looking at. So that gives us a confirmation that our sequence that has been inserted in the cells have the correct, the plasmids that we have inserted have the correct sequence we require or not. So finally, to just summarize what are the topics that we looked at today. We looked at the biomolecules and the processes that are being done in the cells. So we have two important biomolecules that are proteins and nucleic acids. And the important processes that are precursors to protein synthesis is DNA replication, transcription, and translation. And then we talked about different parts in synthetic biology, which are the promoter, the RBS, CDS terminator, and we saw how each of those parts affect the gene expression and what can we how do we assemble all those parts onto a plasmid. And then we looked at different laboratory techniques that help us to take that design, make that design, and insert that into whatever organisms that we want the proteins to be expressed in, and different techniques that help us carry out all those operations. So yeah, so that was the end of the slides. We can have questions now, or should we have the questions on Slack? Ice.
Can you hear me?
No, I couldn't hear you. Did you say anything?
Okay, I'm back. I think I'm back. Can you hear me?
Yeah, I can hear you.
Cool. Yeah, I think for the interest of time, if we don't have any specific question going on right now, since like Hari has answered a lot of questions during the lecture, we can adjourn and like end the class very soon. But before we hop off, I would like to share with you that like we will upload this recording onto YouTube, and we will send you the link that you can access to this recording by this weekend. I'm in charge of that, so I'll make sure we go through that. And at the same time, we will share all this. We will share the slide that Hari used to present as well, so that you guys have a chance to review by just looking through the slide, and also listen to the recordings, and you can just like do it like at a faster speed or like scroll it over and over as you would prefer. The other thing I would like to mention is that we have a homework associated with each lecture. So I will share the homework link very soon, like into this Zoom, and you can also find that in the syllabus link that we sent earlier. So like in each class we will have like at least the link to the recording, the link to the slide, and also a link to the homework, so that you can use the homework to guide through what we have been discussing about. Let's see. All right, so that's the form for the homework. And you get an email when we have these information upload, but you should also have access to this syllabus file that I just posted. It should be an email that we sent to you before the class start, and we'll keep sending this over to you over and over so that you don't lose it. The links to each slide or recording should be in this syllabus document. And if you are lost, you cannot find anything, please come back to this link. For the homework clarification is that like we will grade the homework roughly 1 week after the class. So we'll try to grade and send it back around the day that we have the class of the week after. But if we were busy and didn't get there, please bear with us, and we will get back to you as soon as possible. If you didn't get it done in time, you are also allowed to work through the homework later, but it may not be graded timely. So there's if you do it in time, you get response in a timely manner. If you don't, you will still get response, but we can promise that we will get there by which time, because we have to focus on the homework that was submitted on time first. And finally, before I let Harry say anything before the end, I would like to apologize to those who have troubles logging into this Zoom. We didn't expect the participants number to exceed 100. And I see that emails coming in and what can I do? So we work on our end to resolve that, and I believe it's now resolved. In next lecture, we should have capability to host up to 500. So I don't think we will exceed that number. Last year when we do beginner class, we have around 50 people attending live class, but now we have more than 100, and we were not expecting that at all. Thanks everyone for the interest. So well, we'll stay in touch. Thanks, Abika, for letting us know. I will generate the new link and send that over through an email. And that should be better than trying to get it done right now. So yeah, I think that's all for me. Maybe Harry or Kato, if you have anything to share, please go ahead.
Okay. Can you hear me?
We can hear you.
Okay, thank you so much for today's lecture, Harry, and thank you so much whoever attended this lecture. I am Sebunya Kato, and I am very happy that you have attended today. I have shared the link to our website in the chat. Maybe I can share it again. For any information which you need, I saw people asking how they will access the links. We have shared you the links to the syllabus in the email, but also we are going to upload most of the things on our website. And also when the recording to this lecture, when it's available, we shall upload it to our YouTube channel. I wish to request you, those ones who have attended today's lecture, please, you can inform your fellows in wherever country you are. You can share with them information about these lectures. The registration is still open. They register and join us to study more. Next week, we shall have another lecture at the same time, and it will be diving deep into the core of synthetic biology. Please don't miss that lecture. Endeavor to attend. Thank you so much, Harry, and back to you.
Yeah, I know that I think I was a bit fast in covering all the topics, and there were a lot of topics to be covered in a short time. So I completely understand if I was not if you did not understand the slides, or if I was too fast. So if you have any questions related to the slides or any other topics, please feel free to message me on Slack, or we can also have the discussion in the new channel that has been made in the Slack, so that all the questions can be answered and we all are on the same page. And the further the lectures in the next weeks are also going to be very interesting, and it will be very good if you clear up all the doubts and have all the basics cleared up before we move on to the next week's lectures.
All right. Well, I just checked the access, so you should be able to access the homework form appropriately. For the Google form that we have that we have the how do we call it? The syllabus. You should be able to see the syllabus, but we are not granting access to everyone because like it was used for us to you know, like to update the links so that like it was not edited and we have to come and fix it all the time, just in case there are some mistakes going on. So hopefully that should work. And if you have any questions or like any suggestion for the class, please feel free to reach out to us via email and we'll be happy to talk over. Yeah, I think that's it that I would like to share. Please also visit our Slack, and we have like Slack channels to talk on the homework questions or also the lecture questions that we can use that for our better purpose. I think that's all on our end. I think we can say goodbye for today, and like we'll see you again next week. Hedy will be the instructor for next week, I believe. Thanks a lot, everyone, for joining in.
Thank you.
Okay, bye-bye, and have the rest of your evening or day or morning. Let it be good. Thank you.
Goodbye. I'll stop the recording now.